Get going. Okay, so we left off last time here. Did we leave off here? Yeah, I guess I had mostly finished it, huh? Okay. So, anyways, where did I stop? Right here, entering the H pluses in. The two-sided chromes, the positive electrons. We had negative charged copper and iron here. And then we built our peroxide bridge. And then H pluses are going to come on those peroxide bridges right here. Remember, they'll have lone pair, a bit of negative charge associated with them. And so the H pluses will jump on. That will cause the breaking of this bond, or vice versa. The bond could break first and then draw H pluses in. It's a very unstable situation, so peroxides are quite unstable, actually. What's going to happen next is once we have those H pluses on, broke our peroxide, that would give us essentially two OH minuses that are bonded to metals. Okay? So they're kind of like partial OH minuses. Remember, those OH minus part comes from the cytochrome Cs, the two extra ones. That's our total of 4, site C. And so that's going to bring on the two new H pluses here. This is going to cause these to be released as H2Os, which again are neutral, right? And so can release now into our water. And again, this is going to then reset right here. Okay. Anybody have any questions? Fantastic. Where are these H pluses coming from, by the way? Which side is the bottom here? It's the matrix. Okay, mitochondrial matrix. Okay, so this is the two metals forming the peroxide bridge in your cytochrome C. And again, here's the iron in the heme. This one's that copper right there, stabilized by histidines. And so what you'll see here is, again, we have the iron 3+, copper 2+. These are modulating that energy for the peroxides to make it lower energy than it would normally be, but it's still high enough energy to drive the other reactions. Okay? Everybody okay with that? Fantastic. All right, so again, 4 chemical protons are going to reduce O2 to the 2 H2Os. Remember, that's that peroxide bridge getting broken and then added onto releasing the 2 H2Os. And so this right here is the 4 chemical, the 4 electrons are going to be added to this. This is going to be the 4 H pluses coming in. These are the chemical protons. Then you have 4 H pluses that are the pumped protons. Those are the ones that are going to be pumped from the matrix to the inner mitochondrial membrane, or intermembrane space. Anybody have any questions? Good guy. Notice, by the way, the color combinations here really do a lot of work, okay? In case you're forgetting things. Green into the intermembrane space, the cytochrome C going from reduced to oxidized here, red to blue. Notice again, these went red when they were reduced. When they get the oxidized forms, they go to the blue, right? Okay, so the electron is going to flow via 2 pathways through the electron transport chain. And we can actually see these pathways right here. And so again, this is the electron transport chain. This is the matrix, intramembrane space. These are your joinders with the citric acid cycle. We have the NADH here, FADH2. Those are joined into the complex 1 and complex 2. Remember, this is not just a superficial joining, okay? This is overlapping of the enzymes, period, okay? So these first 2 complexes, they're going to use your Q pool, and that Q pool is then going to then give you the QH2s that would feed into the complex 3. And remember, the complex 3 does that use part of a QH2, recycle part of it to another Q, right? It does that twice. This is going to give you your cytochrome Cs that are right here. These are going to go into complex 4, providing that energy. And that is going to be where we actually get the oxygen going to H2O. And again, each of these, complex 1, 3, and 4, are going to be pumping protons. What about complex 2? Is that pumping protons? No. Okay. So, respirosome, this is a massive complex in humans. This consists of 2 copies of complex 1, complex 3, and complex 4. So right here's the complex 4s. You can see them on the left and right shoulders. Everybody see those okay? It kind of looks like a person, doesn't it? See the shoulders, body, head, arms. Okay, so complex 4s, shoulders. We have this outline right here, so we can see through it to the complex 4 here and get a better idea what the hell's going on. Okay? This is the cytochrome C, by the way. This side is the matrix side. Okay? This is the intermembrane space with the cytochrome C. And so this is the complex 1 portions right here. They are the arms, if you will. And then the body right here is the complex 3. And remember, there's 2 copies of that. And it's hard to see those 2 copies because otherwise they'd obscure everything else. But they're sort of the front, back. Okay? So this structure is going to allow for the complex 2 to associate a gap between the complexes 1 and 4. It'll enhance efficiency because remember that many of these things would have to travel, could get lost, could take time to travel, and then you also have a number of fairly highly reactive intermediates in this, right? And so this will cut down on those going off and reacting with other stuff and causing issues, okay? Though interestingly enough, most of your degenerative diseases, the ones that get really bad as you age, are actually mitochondrial type, okay? So for example, do you know what the largest determinant of like neurodegenerative diseases or Parkinson's or Alzheimer's or any of those are? Mitochondrial. It's very likely they're mitochondrial. But that's because all your really reactive stuff usually happens to mitochondria anyways, right? So, the partial reduction of your O2 is going to generate highly reactive oxygen derivatives. These are called reactive oxygen species, or they're abbreviated ROS oftentimes. The ROSs are implicated in aging and a growing list of diseases. ROS can include the superoxide ion, the peroxide ion, and the hydroxyl radical. By the way, the vast majority of oxidation in nature happens via the hydroxyl radical. So like when you're talking about the oxidation of something that's sitting around in open air, most of it happens through the hydroxyl radical, okay? Not through oxygen, O2, which is what people usually think, right? No, it's the hydroxyl free radical. Interesting little side note to that. As it turns out, that raindrops hitting stone or a stream hitting stone actually generates hydroxyl free radicals. They're way easier to generate than everybody ever thought. It's pretty recent stuff, by the way, like last 4 years. So the cytochrome C oxidase, this does not release the ROS by holding O2 tightly between that Fe and Cu ions. We kind of talked about that before. That peroxide bridge being established is both to make it more reactive and less energy barriers to overcome, normal enzyme stuff, but also to protect you, right? Because if those things were released, they would react with just everything in your body, okay? You would get hundreds of intermediates and possible side products, okay? As you learned from the free radical reaction that we did in lab, where we had to put it on ice, right? For the organic peoples. So the pathological conditions that may entail free radical injury are going to be these ones right here. Parkinson's disease being one of the more, crazy ones, diabetes, renal failure, anyways. And this list, basically, this is a 2012 list right here. This list is much longer now, okay? Or the ones that have likely ties to this is much, much longer now, right? But they only get compiled every now and then because you can't just have somebody sitting there doing this all day. They have to do some research too. All right. So the superoxide dismutase, this is SOD. This is going to be an enzyme that will scavenge superoxide radicals by catalyzing the conversion of two radicals into hydrogen peroxide and molecular oxygen. And so right here we have our radicals reacting with H+. This is the superoxide dismutase. And we get O2 in H2O, which is the hydrogen peroxide. Eukaryotes actually contain two forms of SOD. We have a manganese-containing version located in the mitochondria. We have a copper and zinc-dependent cytoplasmic form. Interestingly enough, the two supplements that seem to have some of the best effects for mood elevation, anti-aging, disease prevention, keeping you healthy enough to fight off diseases. Anybody know what those are? They're manganese and zinc. Great. So exercise is going to be associated with the increased SOD expression. And this is, again, your leading likelihood of developing diseases is actually largely determined by, number one, genetics, nothing you can do about it. But number two, exercise. Do you exercise? How much do you exercise? Do you have a lot of standing muscle mass? Do you have a large amount of fat mass instead? Okay. The functions near diffusion limit rate. So this is about perfect for an enzyme because it's nearly diffusion rate, right? This is the superoxide dismutase mechanism. We have two phases here. We have the oxidized state. It's going to then take on an O2 free radical, anion. This is going to give you an O2. This is going to give you the reduced state because we took an electron away, right? Now that reduced state is then going to encounter a second free radical. This is where we'll take on two H pluses. We'll get the H2O2, which is the hydrogen peroxide. And that's where we take away the extra reduction from this. Now notice that does mean we actually have reduced these oxygens, haven't we? See that? We're actually taking the second pair of O2s and we're giving them one more reduction step. And those are those hydrogens right there, the two reductions. Okay? All right. So catalase, this is ubiquitous heme protein that's going to catalyze the dismutation of hydrogen peroxide into water and molecular oxygen. And so right here we have hydrogen peroxides, two of them. Hydrogen peroxide is not exactly not reactive with humans, is it? Would you want to drink hydrogen peroxide? No, that's a bad idea, right? So what we do is we have catalase, then is going to take care of the next one, that's a poison still, which is the hydrogen peroxide. And so this is going to give you O2 and two waters here. This is going to function again at near diffusion limited rate, and so it's basically a perfect enzyme, as it were. Really, the only limitations and problems here is the fact that, number one, diffusion does not mean it heads directly to one of these. Diffusion means that it bounces around, right? And number two, you may not have enough of these two enzymes to counteract everything that's being released and generated. So you may get overwhelmed at points. So in a protein environment, the rate of electron transfer decreases by a factor of 10 for every 1.7 angstrom distance between the electron donor and acceptor. For groups in contact, the electron transfer reaction rates are going to be approximately 10 to the 13 1/2 seconds. That means that happens 10 to the 13 times every second, right? Or another way of saying that would be hertz. Okay? For the electron transport chain proteins, the electron carrying groups are going to be typically separated by 15 angstroms beyond their van der Waals contact distance. So your rates are going to be approximately 10 to the 4 times per second. Okay? Notice that a relatively modest difference in distance has a very large effect, right? See that? Yeah. And so this is like every electrical reaction or magnetism reaction or any of these things. The distance, as you go further distance, the speed, the magnetic fields, the strength of magnetic fields, all those things drop off exponentially, right? Very, very, very quickly. Okay, so the proteins are going to dramatically improve the rate of electron transfer between the atoms at a distance. If we didn't have these, it would be a big, big problem being out of van der Waals contact. So again, this is a log 10 rate of electron transfer. On these things like electron transfers, by the way, binding, all those things, if it's not a logarithmic scale, then something's wrong, okay? You're really looking at something weird. And you should really pay attention to what they're telling you, because likely they're doing that to hide something. Just saying. More often than not, it's used to sell you something you don't want. Okay, and so the rate through the vacuum would actually be right here, and this would be nothing interfering with that electron transfer, either positively or negatively. Okay, this one right here is the rate through the proteins. And because we have specific evolution paths that have been developed to aid this process, what you'll see here is that the distance that you can cover at the same sort of rate actually greatly improves, doesn't it? Yeah. So we about double the distance that you can cover at the same rate at any given time. Okay. Notice again, though, that the van der Waals contact points, if we're at that short a distance, that electron transfer is incredibly fast. Protein is not going to do anything. You won't have room for a protein anyways. So the flow of the NADH to O2 is going to be an exergonic process. Your NADH plus 1/2 O2 plus H plus. This is going to give you water and NAD plus. And this right here is a delta G naught prime of negative 220.1 kilojoules per mole. Everybody okay with that? Okay. That tells you that would be enough energy to sort of like burn you, right? But luckily it's in redox reaction, so we're not getting that transferred just as heat. Okay? We're joining that with other things. So the synthesis of ATP is going to be an inorganic process. Everybody remembers inorganic, right? We have to push energy in all the time. Is this the same thing as heat, by the way? Yeah. Exothermic, endothermic would be the heat only, right? This is both the delta H and delta S. Okay? This is your delta G's. So again, this would be non-spontaneous. This is actually where that energy is ultimately going to, right? Through our electron transport chain or via our electron transport chain. The ATP synthase, commonly called complex 5, is going to be the molecular assembly in the inner mitochondrial membrane. This is going to carry out the synthesis of that ATP. It's the one that's using up that H plus energy for us gathering all those H pluses to generate the ATP. You may have been going, hey, Dr. Hawson, you're not getting that. The hell, man. Well, there you go, okay? Your complex 5, again, that is the ATP synthase. The name actually tells you a lot, doesn't it? Okay? It's an enzyme that does what? Synthesizes ATP. And it does it after complexes 1, 2, 3, and 4. There you go. So the chemiosmotic hypothesis, this proposes that the electron transport and ATP synthesis are going to be coupled by a proton gradient across the inner mitochondrial membrane. So protons can be pumped across this membrane as the electrons flow through the respiratory chain that we had talked about before. This is again the outer mitochondrial membrane. This one's the inner mitochondrial membrane. And we have the intermembrane space where the positive charge is going to build up. And we have the matrix where we have been pumping H pluses out. We have not been pumping their counterions out, though, have we? And so we have built up negative charge. And again, it's just like osmotic pressure. It's just that it's chemiosmotic pressure because it's a pressure differential created through via charge, right? Same sort of thing you'd see in a battery. So the proton motive force, notice this is slightly different than the electromotive force. Can anybody tell me why this would be different? Are we going to be transferring electrons at the end of this? No. So normally in electricity, we transfer electrons, don't we? Okay, and that's the electromotive force. This is the proton motive force because we're transferring protons. Okay, this is why your book's so damn good. Like every other biochemistry book I've ever read does not get that shit right at all. The energy-rich unequal distribution of protons across a membrane. It's going to consist of a chemical gradient and a charge gradient that powers the synthesis of ATP. And again, that's the proton motive force. They even have the symbol here right, which is just fucking crazy. I've never seen a biology people get this right, ever, except for this book. Only time I've ever seen it. Delta P, the lowercase p, that's italic, normally dictates proton, right? And so this is a change in proton gradient, isn't it? Which is exactly right. Your chemical gradient, again, is going to be delta pH. Your charge gradient is actually, again, correct, by the way, which is the delta psi. And it's just amazing they got that right. And delta pH, literally concentration of H pluses with the negative log of that concentration, right? So you can measure it easier. All right, so when the vesicles were exposed to light, ATP was formed. And so this is the bacteriorhodopsin in the synthetic vesicle right here. And here we have light being exposed. And we have a H plus coming in. When H plus came into this synthetic vesicle here, we built up a charge gradient, right? And so we're basically... By the way, did you notice that this is essentially the same sort of system that like a house would have for solar panels? So we expose it to light, it builds up a charge in a battery, and then that's used to store energy. That's actually pretty clever. So we build up the charge H+, that's then going to power this ATP synthase. The ATP synthase is going to join ADP and orthophosphate, right? Creating our ATP, our chemical energy storage. Notice again, this actually shows you that the respiratory chain and your ATP synthase, these are biochemically separate. They do not actually happen, they don't have to happen with each other at all, okay? You can completely separate them in every way. The only thing joining them is the proton motive force. They're two different pieces, just like solar panels and a battery are two different pieces. Okay. The ATP synthase is going to resemble a ball on a stick. You ever remember? Have you guys ever played that stupid game? There's a ball and a string on a stick, and you try to catch the stupid ball, and it's horribly unfun, right? Like. They give it to you, and 5 minutes later, you're bored and don't want to play with it. Okay, that. Okay, that is an ATP synthase. That is exactly an ATP synthase. It looks exactly like that, and it works kind of like that in the first place. The F0, which is your stick, actually, component, this is the one that's embedded in the inner mitochondrial membrane. It contains the proton channel. The F1 ball component is going to protrude into the mitochondrial matrix. This is going to contain the catalytic activity. And so this right here is what that looks like. Right here. And so we have here our stick. This is going to be where the protons actually come through. And it's basically just a channel. Okay? And right here's our string. See the string? Ooh, ah. Yeah. And right here's the ball, right? Okay. Now this ball is in fact closer to a like turbine or a spinny thing on the end of a stick. Okay? Which I guess ball still works. You could have the spinny ball, I guess. Anyways, F1, F0 through the membrane. Again, this is going to be extending into the matrix with the F1. The F0 is the one that passes the proton gradient through the membrane back into the matrix. Remember, in this case, we're going to be going from the inner membrane space back through to the matrix, okay? Technically, you could actually reverse this flow, but it would not work out well. It'd be much more difficult. So the F1 subunit, this is going to consist of 5 types of polypeptide chains: the alpha 3, the beta 3, the gamma, delta, and epsilon. Alpha and beta subunits are going to be arranged in a hexameric ring. Both are going to bind nucleotides. Only the beta subunits are going to be catalytically activated. So keep that in mind, that we have the alpha, beta, see those in here? This is that hexameric ring. And we're alternating alpha, beta, alpha, beta, alpha, beta. Okay? You guys have all seen a plane prop or a turbine? No? Yeah? Okay. Yeah, wind turbine's fine. Okay. I mean, there's one right out there, right? Okay. Okay. So that also has alpha, beta. Okay? It has the active alphas, which are the props, right? And it has empty spaces, which are the betas in that case. Okay, which are not active, right? And part of that's, you need that differential so that you can actually cause turning action, right? Now, the central stalk is going to consist of the gamma and epsilon proteins. These are the gamma and epsilon. They're the central stalk. Notice, by the way, the central stalk is important. It's the drive shaft, but what else is on a drive shaft? If you just had a pole and that's all you have, it doesn't have any protrusion off it, does it again do anything? No. We need a protrusion off it, right? And so that's the epsilon right there. The gamma subunit is going to break the symmetry of the alpha 3 beta 3 hexamer. Each of the beta subunits is going to be distinct by virtue of its interaction with different asymmetrical face of the gamma. And so we can't see it here because this one's from the side. We'll see it better in the next figures, okay? But this gamma is actually going to break that symmetry. You can kind of see it, but not well. This was that asymmetry, by the way. You see the asymmetry? Kinda. Maybe. See the little gap? It goes like that. Okay, so the F0 subunit, this consists of a ring that's going to be comprising from 8 to 14 C subunits that are going to be embedded in the membrane. A single LA subunit binds to the outside of your ring. The F0 and F1 subunits are going to be connected in 2 ways. We have the central gamma epsilon stalk, which we can see right here. That's that drive shaft, if you will. Everybody okay with me saying drive shaft? The exterior column is going to consist of 1 A subunit, 2 B subunits, and a delta subunit. And so this right here is that right there, the exterior column. Okay? And again, it's like a car, isn't it? Or like a wind turbine. Is there just the drive shaft and nothing else in the wind turbine? Hell no. There's the pole that it sits in, right? Yeah, that's there too. So there's the connective tissue, if you will. Okay, so the mitochondria, ATP synthase, is going to form homodimers. This will stabilize the individual enzymes to the rotational forces that are required for catalysis. This facilitates the curvature of the inner mitochondrial membrane. And so right here we see the matrix. And again, remember they have these protrusions into the matrix, right? Remember that? So it protrudes into the matrix. This is one of those. This is that inner membrane space then. And what you can see here is that, again, these are going to be attached and they're going to form homodimers. Homo meaning same, right? And so just 2 dimers that come together. Now, because they're linked like this, where you have these linked, when these are rotating, what happens when they rotate? Anybody ever been here when it's windy in this room? Been here a couple times when it's windy, right? What do we hear when it's windy? It's really loud, right? Very unpleasant, okay? Why does it make that sound? You ever looked up at the turbines while it's windy? Yeah, well, it's not just going fast, it's vibrating. It vibrates the turbine. And that's because, yeah, if this was perfect, it would not vibrate, right? But is this perfectly symmetric in every way whatsoever? And their environments are symmetric so they don't hit anything? No. No. And so what happens is the rotational force actually does ejection force and would vibrate these, right? And that would tear that right out of the membrane space. Remember, these are just van der Waals forces, aren't they? So you link these two together, their rotations are actually going to counter each other. And so that vibrations are going to start countering each other. It's actually the same way those oil rigs stay afloat and work. Okay? You ever seen the oil rigs? Yeah. Similar mechanism. You can also do the same thing with like wind turbines as well. And you can have a counter force that's going to stop that. Same way the elevators work, by the way. It has a counter force that stops it from vibrating a bunch when it tries to move. Otherwise, every time it moves, you know how it goes dink. That's because our counter force isn't super great, but yeah. Okay, so the Christea, the formation, is going to allow the proton pumps to localize the proton gradient in the vicinity of the syntheses, which are going to be located at the tips of the Christea. And so what you're going to see is that typically these are located at the tips of the Christea, and then you're going to have your complex 1 through 4s located on the body. And this is going to build up concentration within this Christea. See that? And you'll have less concentration out here. You'll still have some concentration, but it's going to drift downwards as you go out here. Anybody have any questions about that? Okay. Wait a second. Are membranes solid? No, this is mobile, isn't it? Okay. Why is this concentrating out here then? It's the shape. So the shape of it actually is helping to create the Christea in the first place, because the membrane is associating with it, right? And so it's like putting little clips on your pants, right? They're going to start pulling the pants on it. Okay? Is that making sense? Okay, so again, that'll enhance your efficiency, your ATP synthase. The ATP synthase is going to catalyze the formation of your AT from the ADP and the ortho-free phosphates. And this right here is that overall reaction. By the way, these are with the charges, which you don't normally get to see, which is kind of interesting. Okay. The actual substrates are going to be ATP and ADP. These are going to be complex, though, with magnesium 2+. And so magnesium 2+ is absolutely vital. And again, one of the reasons why, if you're feeling tired, guess what supplement works really well? Magnesium. What is that ad that's everywhere? Magnesium breakthrough? Is that it? Damn thing shows up all over the fucking place. All right. Maybe that's just for me. Probably my age group, right? Yeah. Usually there's supplements that you take, like drinks and stuff, or I guess they could probably do it in spray form. That's interesting. I think the one they're giving me is like a pill, I think. But yeah, that's funny. Probably our age groups, right? Yeah. So a terminal oxygen atom of the ADP, this attacks the phosphorus atom of the orthophosphate. It's going to form a pentacovalent intermediate that will dissociate into ATP and H2O. This right here is the ADP. This is the orthophosphate. You're going to get an attack on the phosphorus here, and you need an H+ to finish this reaction. So this is going to be the pentacovalent intermediate right here. Notice why it's pentacovalent. There are 5 bonding oxygens, aren't there? Okay, which is why you need the H+. So you attack this phosphorus. Again, that phosphorus will be the one that would have that positive charge. This has that slightly negative charge. That's a normal sort of SN2 reaction, right? And then you're going to get an H plus coming onto this excess oxygen, the one in the green. That will leave as water, which is an external leaving group, and that will leave this in the normal ATP form. Notice that in terms of organic mechanism, we're actually kind of skipping a few steps, right? We're just looking at the overall thing. Like, we didn't even talk about why this reestablishes that double bond. But we don't need the entire mechanism for biochemistry, right? The isotopic exchange experiments, these actually revealed enzyme-bound ATP forms readily in the absence of proton motive force. The APT does not leave the catalytic site unless the protons flow through the enzyme. So interestingly enough, this part here actually works okay without the proton motive force. But they don't leave the site, and so you can never progress. The part that actually needs the proton motive force is actually throwing the damn things out of the enzyme. Okay? So ADP going to ATP. If you actually use a labeled water, then what you can do is you can have this label when it goes backwards to the ADP going back to the orthophosphates. And you can actually see them break back apart. So the F1 subunit of your ATPase, this contains 3 beta subunits. There's going to be 3 active sites on each of these enzymes. The binding change mechanism states a beta subunit can perform 3 sequential steps in ATP synthesis by changing the conformation. I did get everybody realized that I posted that video for that problem. Okay. So if you go on to the playlist, that problem's up there. I had to do it in 3 parts because I kept getting interrupted, as you can see from my calls and stuff. God. Step 1, ADP and orthophosphate binding. Step 2 is ATP synthesis. Step 3 is going to be ATP release. So this right here is actually this process. And you can see these different steps in this process. Remember, part of this process is actually going to be the gamma moving. That gamma moving is going to be forced by the proton motive force. Okay? That's the force behind this gamma moving here. Okay. Notice the gamma has 2 parts that are asymmetric. We have a protrusion right here, and over here we have a empty space. Everybody say it okay? What does that look an awful lot like? A gear assembly. Yeah, same sort of thing a human would use, right? Okay. And so this empty space, this is in the beta 3 position right now. This is going to be the O version of it, okay? And this is going to be where we have the ADP and the PI, the orthophosphate, coming together in the open position. This right here is going to have the L position. This is going to be where we have the ADP and the orthophosphate. They're in that position already, and they're anchored down in there. And then this is the beta 2, which is the T position. This is the one with the protrusion that's going to be changing that. And so that one is going to have it where our ATP and our ADP, or the orthophosphate, these are going to be in equilibrium, and they're going to be doing their reaction. Okay? And remember, it's going to go from, the pattern will be the TOL. Okay? If you happen to be starting on O, it would be OLT, right? If you happen to be starting on L, it would be LTO. Okay. And so we're going to repeatedly go around like this. Click, 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 click. L again is the loose conformation. This binds your ADP and the orthophosphate. You cannot release the bound nucleotides once you've bound them. T is going to be the tight conformation. This is going to bind ATP with great affinity. This is going to convert your bound ADP and orthophosphate to your ATP. Again, that's the T right there. This right here being the L. This cannot release your bound nucleotides still. The O, open conformation, this can bind or release your nucleotides. So this is the one where we get release of the ATP, we get binding of new ADP. Okay? This is the only one that will allow the release. This one, they're bound. Remember, you actually take energy, you need the proton motive force to release these. It's not actually the reaction that you need the extra energy to do. And then this one right here is the form where you have a great affinity for ATP, and this is going to help you along for your ATP. Okay? It's going to bias that equilibrium. Now, the rotation of your gamma subunit is going to drive that interconvergence of these three forms. It's going to provide the energy to keep you going along this pathway. Okay? It's your click, 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 click driving shaft, right? All right, so each subunit is going to progress from your, again, TOL, TOL, TOL. No two subunits are going to be in the same conformational form because, again, we have three different spots on our gamma. We have one that has the O open, one that has the T tight, one that has the loose, right? And that's going to move in this form. Okay, so the cloned alpha 3 beta 3 gamma subunits are going to be attached to a glass slide. The gamma subunit was then linked to a fluorescence labeled actin filament. This provided a segment that could be observed under the fluorescence microscope or microscopy. The ATP addition caused the actin filament to rotate in a counterclockwise direction. This is visually confirmed that the gamma subunit was rotating, driven by the hydrolysis of your ATP. The hydrolysis of a single ATP powered the rotation of the gamma subunit, 120 degrees. How many times do you have to rotate to get a full 360? 3 times. How many states are there? 3 states, yeah. So each rotation gives you a different state, right? This is what that looks like. This is, again, we just took the ball portion, right? That's the only portion we want. We want to see what this does in the presence of ADP and ATP, right? And so our actin filament is attached. We feed ATP. We get ADP out, right? Why would we get ADP out? Which is higher energy? ATP, right? So without the proton motive force, well, adding ATP to this is going to drive this in the opposite direction, isn't it? Yeah. And so we're driving this the opposite direction we would normally do when we have the proton motive force, right? And so ATP causes this to go counterclockwise, and thus we get the ADP out, okay? Do you know what would happen if you left the battery connected to a wind turbine and you had nothing to disconnect the battery when there's no wind? Okay, you guys ever worked on like an electric vehicle, electric motor at all? You guys have an electric vacuum, one of those battery-powered vacuums, like a Roomba? Okay. Have you ever had it off and then you start pushing it around with your foot? What happens? Light. Flicker, 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 right? Why is that happening? Because electron motors, when you provide them electricity, turn one way. When you run those motors without providing them electricity, guess what happens? You start charging the battery. So what you're actually doing is you're generating electricity when you're pushing your vacuum around with your foot. And it doesn't really have much where they basically use that to either power the battery, or in the Roombas, it appears they just let it discharge into the lights. Yeah. I don't know if that's on purpose or by accident, but yeah. By the way, did you know if you use a fan and you have a stronger wind that blows into the fan than the fan generates, you can actually fry the fan? Wow, huh? Yeah. And that's because it will actually generate electricity if you push it the other way. Okay? If you start pushing it around and instead of it pushing stuff around. Okay. Anyways, we're doing that. We're basically using this the opposite way, right? We're discharging the battery instead of charging it. Is that making sense? Okay. Okay, and we're seeing the rotation right here, and this actually powers that rotation right here. And so it's kind of interesting. We got to play around with the motor a little bit separately, right? The proton flow is going to occur through the F0 component of the ATP synthase. The stationary A subunit is going to butt the membrane spanning ring formed by the C subunits. The A subunit is going to contain two hydrophilic half channels, each of which interacts with one C subunit. One opens the intermembrane space, one opens the matrix. The membrane spanning ring is going to be formed by 8 to 14 C subunits. The vertebrates will have 8 units. Each polypeptide chain of the C subunit will form a pair of alpha helices. These will span the membrane. The glutamic acid or aspartic acid residue is found in the middle of one of the helices. When unprotonated, the C subunit will not move into the membrane. When protonated, the C subunit will move into the membrane as the ring rotates by one C subunit. And so this is actually what you're seeing. So this right here is your C subunit. Here we have the aspartic acid. This is the half channel right here. So the intramembrane half channels. This is the matrix half channel. And again, we have the subunit A right here. And so what you're literally seeing is as we bind a proton on here, we're going to, so we get this in, it binds the proton on here. This is going to cause this to move, okay? And it will move one rotation, one movement of a C subunit. And that right there is what causes then our crankshaft to then move later, right? Okay, which we haven't actually gotten to that part yet. So the rotation of your C ring, step 1, the proton is going to enter your half channel from, again, the proton-rich intramembrane space right here. This is the intramembrane half channel right there in your subunit A. The proton is then going to bind to a glue or asp residue in the C subunit, which is right here. Okay, are you okay with that so far? Why is it aspartic acid or glutamic acid? If you're proton-rich, what would that mean for your pH? Lowish, right? And so you want something with a relatively low pKa so it can have protons pop on and off of it, right? Something that's pKa is going to be approximately in the region of the pH, right? We're actually seeing a buffer interaction. So the ring, the C ring, is going to, step 3, rotate clockwise, one C subunit at a time. Hey, this said counterclockwise. What the hell, Dr. Clausen? What's going on? Was this the proper direction for the activity that the mitochondria would normally do? Does the mitochondria ATP synthase break down ATP? No, we want the opposite direction, don't we? Yeah. So clockwise. So again, one C subunit at a time. Step 4, the proton is going to exit the half channel to the matrix. This is going to be followed by a complete rotation of your C ring. And so now this pops off in the opposite direction, going through the matrix half channel, which will then exit it from the matrix. Remember that this rotation is actually what's causing this to be able to do that, right? And so this actually ends up being monodirectional, doesn't it? Yeah, because it popping on rotates it so it matches up with the matrix half channel, which case it's osmotically, it's actually energetically favorable for it to pop off on this matrix half channel and then come off over here, right? And then this switches back up to match the intermembrane half channel, in which case the protons are going to come back on. Everybody see that okay? Okay, so the movement of the protons through your half channels, again, from the high proton concentration to the low proton concentration, is going to power the rotation of your C ring. The rotation of the C rings powers the movement of the gamma subunit. This in turn alters the conformation of your beta subunit and thus powers that process of forming ATPs, primarily through throwing ATPs off when they get stuck on there. Okay? So this right here is that overall process for your C ring portion, your F0 portion. This is your inner mitochondrial membrane, intermembrane space, matrix. And so what we're seeing here is they jump onto the first half ring here. Why is this negatively charged here? It's a deprotonated carboxylic acid, right? So deprotonated gives negative charge. And so that's going to jump on here. At that point, we would have a proton in here, which is right there. And then what's going to happen then is that will cause a rotation when that jumps on there. And that's that rotating clockwise. This is then going to match it up with the matrix side. So we'll flip this negative one over into this part. The hydrogen right here is going to match up with the matrix one. Now, this matrix one is then going to be energetically favorable to release. And a proton can now join back onto this one. Does anybody have any questions on that? Now note that this is not actually a cycle here because we had a different group here. It is now matched up, right? See that? We didn't actually reset. Okay? The next one, it will actually reset. So when it does that twice, it'll reset. Okay, so this right here is, again, the intermembrane space. We get a proton jumping on. This will actually turn this way, clockwise, which will match the matrix, which will let this out. So this actually gives us a 360 rotation of our gamma subunit, leads to the synthesis and release of 3 molecules of ATP. So when we get a full 360 rotation, 3 molecules of ATP are generated. Again, this phosphorylation actually shows a spatial relationship with the components. So again, the proton motive force happens on the side walls of the crystea, pumping into the intermembrane space. This right here, which is the synthase complex 5, actually happens at the tips of the crystea, right? Where we have an extra buildup of the proton motive force. And that's where we get the ATP synthesis. The alpha and beta subunits of your ATP synthase and G proteins are going to be members of the P-loop NTPase family of proteins. The signaling properties are going to depend on their ability to bind NTPs and NDPs. They do not exchange nucleotides unless stimulated by the interactions of other proteins. And so they're actually related by this P-loop that we kind of mentioned before and talked a little bit about. Okay? So the respiratory chain is going to regenerate the NAD plus for use in your glycolysis. The inner mitochondrial membrane is impermeable to NADH and NAD plus. The glycerol 3-phosphatate shuttle, one means of transporting the electrons from the NADH into the electron transport chain. When the cytoplasmic NADH is transported by the shuttle, it's actually going to be oxidized by the respiratory chain. We'll actually end up generating 1.5 ATP, which will be formed. Because the FAD rather than the NAD plus is actually the electron acceptor in this process. Normally NADH would have generated the 2.5, right? Instead, we're only getting the 1.5 because, again, that's the FAD that's actually going to be the one that does that. The electrons from the NADH are going to be transferred to the dihydroxyacetone, I forgot the di, phosphate to form the glycerol 3-phosphate. The step 2 is going to be glycerol 3-phosphate moved into the mitochondrion and reoxidized to dihydroxyacetone phosphate. Electrons are going to pass to the FAD prosthetic group. This is going to form the FADH2. Again, this is that glycerol 3-phosphate shuttle that we're doing. And this is how we're going to get that NADH into the mitochondria, that energy from that, those electrons, so that we can actually do something with them. And they're not just stuck as NADH. Whoa. I don't know what that is, but... so the electrons, again, we have an FAD prosthetic group, or FADH2. That's why the 1.5 ATPs. Step 3 is going to be the reduced flavin is going to transfer its electrons to a molecule of Q. This can enter your respiratory chain in your Q pool as QH2. And so, yes, that is that complex 3 that you guys love so much, right? Okay. That you appreciate. So again, this is this whole shuttle, as we're seeing. Remember, this will be the matrix here. This will be the intermembrane space right here. And so again, this is the glycerol 3-phosphate shuttle that we're going to be doing this whole process. So this here is the cytoplasmic glycerol 3-phosphate dehydrogenase out here. We'll have the NADH, or NADH2 plus, if you will, that is going to enter this shuttle. It's going to do it by transferring its H and H plus to this cytoplasmic glycerol 3-phosphate dehydrogenase by transferring those to the glycerol 3-phosphate. This will give you the NAD8 plus that's regenerated back in your cytoplasm. This right here is going to give you the glycerol 3-phosphate. This glycerol 3-phosphate is going to come here onto the mitochondrial glycerol 3-phosphate dehydrogenase. Again, this is the one that will be in the intermembrane space. This is actually going to have the enzyme-bound FAD transfer those hydrogens onto it. That's where the prosthetic group that's going to actually collect that electron density here. This will then regenerate the dihydroxyacetone phosphate, which can come back to the cytoplasmic glycerol 3-phosphate dehydrogenase and do this again. Okay? And so you can kind of think of it literally as a payloads getting transferred from the shuttles, each shuttle. And the payload here is, hydrogens, but specifically, more importantly, the electron density, right? The electrons that accompany them. And so we get the FADH2. This is going to transfer. Excuse me. Yeah, wow. That was really... using the mitochondrial glycerol 3-phosphate dehydrogenase, this is going to transfer that electron density onto Q, giving you the reduced form of Q in your Q pool, which is now in that membrane, intermembrane, that we would do all these reactions anyways. And so that has joined the Q pool now, okay? Now, because it joined right there in the way it did, that only is going to be able to generate 1.5 ATP, or 1.5 ATP. By the way, this is that overall relationship for the shuttle in a more easy to remember format, okay? And so in the cytoplasm of the cell, we have NADH and H+, and in the mitochondria, we have the enzyme-bound FAD. This transfers the electrons to that. In the cytoplasm, we'll end up with the NAD+. The mitochondria will end up with the enzyme-bound FADH2, which goes to our Q pool, right? So the next shuttle that we need to discuss is the malate aspartate shuttle. Have you guys heard of the malate aspartate shuttle? Just a curiosity of how tightly this is bound to all the other classes? No? That's almost disappointing. So the malate aspartate shuttle, this is going to transport electrons from the cytoplasmic NADH into the mitochondria. This will form mitochondrial NADH in the heart and liver. It's going to be mediated by 2 membrane carriers and 4 enzymes. So the malate aspartate shuttle steps is step 1, the electrons are going to be transferred from NADH in the cytoplasm to oxaloacetate, forming malate and NAD+. Step 2, the malate traverses the inner mitochondrial membrane in exchange for alpha-ketoglutarate. Notice, by the way, we're transferring very important intermediates that show up in a bunch of different metabolic pathways, right? Step 3, in the matrix, the malate is then going to be reoxidized by NAD+. This will form oxaloacetate and NADH to form NADH. We've regenerated our NADH back inside our mitochondria, right? Within the matrix. Okay? So we transferred the energy from an NADH outside the mitochondria into the matrix of the mitochondria. The glutamate is going to donate an amino group to the oxaloacetate. This is going to form aspartate and alpha-ketoglutarate. Step 5, aspartate and alpha-ketoglutarate are going to enter the cytoplasm. Step 6, in the cytoplasm, oxaloacetate is going to be regenerated, and the cycle will be restarted. And this is why it's called the malate aspartate. Aspartate on the cytoplasm side, malate on the mitochondria side, right? And here we get that right there. So this is again going to be that malate aspartate shuttle, but now we have a nice, beautiful figure to talk about it, right? And so we'll start here, the NADH and the cytoplasm. And this is going to then give us the malate on oxaloacetate. Okay, so oxaloacetate is going to take on the energy from the NADH, and this is then going to go into the intermembrane space, from the intermembrane space into the matrix. The malate will then transfer its electron density off onto an NADH there. This will go to oxaloacetate. The oxaloacetate will exchange with glutamate. This will give us an alpha-ketoglutarate and aspartate. I'm going to keep following the left though, and then we'll talk about the right portion. Okay, so the alpha-ketoglutarate is actually what's being exchanged for malate. Okay, and so this is exchanging malate for alpha-ketoglutarate for this channel. The alpha-ketoglutarate is going to be regenerated to oxaloacetate in the reaction that takes aspartate going to glutamate. Okay, now this side of that channel. So we're at the point here where we've went alpha-ketoglutarate plus aspartate gives us oxaloacetate and glutamate, right? And the oxaloacetate is going to be what is going to take NADH to NAD+. Now, this has to have that matching glutamate moving around. So our matching glutamate is going to go through our channel. This is going to be what exchanges for aspartate, and how aspartate is going to move back through that channel back into the intermembrane space and eventually the cytoplasm. This right here is going to again be the exchange of the aspartate for glutamate. Glutamate comes into the matrix. Glutamate is going to be what reacts with that oxaloacetate, giving us the aspartate right here. Now, this is that overall sort of effect here. Again, that's the exchange of NADH for NAD plus in the mitochondria, and vice versa. NAD plus in the cytoplasm, exchange for NADH in the mitochondria. Okay? And this is how you can directly transfer, well, directly, quote unquote. So a whole lot of processes, right? But you can directly transfer your cytoplasmic NADH for mitochondria NADH, okay, or vice versa, right? Depending on which direction you need to go. This is the entry of the ADP into the mitochondria. This is going to be coupled by the exit of ATP. This is going to be by the ATP-ADP translocase. So here we have the ATP-ADP translocase. Another name for that is the adenine nucleotide translocase, or ANT. Notice that's a nice abbreviation for the adenine nucleotide translocase. The specific transport protein that enables the exchange of your cytoplasmic ADP for the mitochondrial ATP. It's going to constitute about 15% of the protein of the inner mitochondrial membrane. That's a whole lot, isn't it? Okay, why is it a whole lot? Do you need ATP in the mitochondria? Is that the only place you need it? No, that ATP is what's powering most of your body, isn't it? Yeah. And so this is your exchange function. This is how you get that ATP back out. And remember, it's not going to diffuse through the membrane, is it? So the ATP and ADP, again, bind to the ANT. Everybody comfortable with ANT? You okay if I say ANT? Okay. This is actually going to do this without magnesium 2+. So its inhibition of the ANT is going to lead to the inhibition of the cellular respiration because you'll get a buildup of ATP on one side and a buildup of ADP on the other, right? And then you won't have any actual reactions occurring because you have too high of a concentration of one of the products instead. And so again, this is the ADP3- in the cytoplasm reacting with the ATP4- in the matrix, gives you that ADP3- in the matrix and ATP4- in the cytoplasm. Is your book half the same one? I could swear I took this from your book. Instead of yields a plus. Anyways, that would be interesting. The translocase is going to contain a single nucleotide binding site that alternately faces the matrix and the cytoplasmic sides of your membrane. Remember, we're having an exchange here. So basically, it's going to be like a little cup that flips. Okay. So step one here, we have the inner mitochondrial membrane right here. We're open for business. And so the cytoplasm, ADP, enters our open site. We then have eversion. This is going to dump that ADP into the matrix where we need additional ADP because we've been doing our respiration, right? So we got ADP in the matrix now. Now we are open to the inside, the matrix. So ATP from the matrix is going to enter this this shuttle, this translocase. Went backwards. This is going to then allow this to do the eversion. And so again, we're going to evert, or we're going to have the eversion occur. And this is then going to have the ATP exposed to the outer portion, and this is going to release that into the cytoplasm. Now, notice that, again, we're not actually resetting to the initial state here, are we? Okay? And so that initial state, that's why that's eversion, evert. Okay? Because we have accomplished something, we don't have the same ADP and ATPs. All right, so the phosphate carrier, this is going to mediate the exchange of cytoplasmic H2PO4- for mitochondrial OH-. This is actually kind of interesting because we actually maintain a charge balance by doing this. So this works in concert with our ANT to exchange the cytoplasmic ADP and orthophosphate for the matrix ATP. The ATP synthesome, this is going to be your large complex composed of the ATP synthase and two transporters. I know we have, we concentrate on parts of larger complexes because they kind of build up into larger and larger and larger complexes, eventually giving you your mitochondria and then organelles and then cells, right? And we continuously build you up. So this is actually your intermembrane space right here. This is your inner mitochondrial membrane. This is your matrix. This is the ATP-ADP translocase, and it's doing exchange of your ATP and ADP, right? ADP, we're trying to build up in the matrix. ATP, we're releasing into the intermembrane space and then eventually the cytoplasm, right? Right here, we have the phosphate. And so this one's going to be the phosphate carrier. We're going to be building up the orthophosphates in the matrix and releasing OH- back into the intramembrane space. This one right here is going to be the tricarboxylate carrier. This is going to be exchanging the citrate and an H+ for your malate. And this is going to be resupplying that matrix with our citrate and H+. I'm sorry. We're going to be dumping the citrate and H+ into the intramembrane space. I said matrix, didn't I? intermembrane space, and getting the malate in the matrix. This is going to be the dicarboxylate carrier. This is going to be taking the malate into the intermembrane space and exchanging it for phosphate in the matrix. Anybody know why this is tri and dicarboxylate? Tri, carboxylates, right? Okay. All right. So the ATP, this needs of the cell are going to be determined by the rate of the respiratory pathways and their components. Approximately 30 molecules of ATP are going to be formed when a glucose is completely oxidized to CO2. 26 are going to be formed in the oxidative phosphorylation that we just covered. 2 are going to be formed in the citric acid cycle, and then 2 are formed in the glycolysis. And so this is why you're at your best energy production when you can freely supply CO2, right? And then the other pathways are sort of emergency measures, aren't they? And then that's that right there, because 26 of those are formed in the oxidative phosphorylation. This is actually the table that sums up everything that we've just been through. And so this right here is those 2 molecules of NADH that are formed in, let's go from the start actually. So this is glycolysis right here, okay? And that's that glycolysis that we talked about a while back ago. And so this was that phosphorylation of glucose. We used up one of the ATPs. The phosphorylation of fructose 6-phosphate, we used up another one. The dephosphorylation, we did that twice of 1,3-BPG, and so that gives us 2 ATPs total for the 2 times we did it. The dephosphorylation of 2 molecules of phosphoenolpyruvate gives us 2 molecules total of the ATP. So the 2 molecules of NADH are going to be formed in your oxidation of 2 molecules of acetaldehyde 3-phosphate. Keep in mind that those are not actually generating ATP yet, okay? They actually have to go through shuttles and they have to go into the matrix, don't they? Okay. And so conversion of pyruvate into acetyl-CoA, this happens inside the mitochondria. 2 molecules of NADH are going to be formed again, but we haven't actually gotten at this point to the oxidative phosphorylation, have we? And so those NADH, they don't actually generate any ATP yet. We'll take care of those later. The citric acid cycle, again, inside the mitochondria, is going to have 2 molecules of adenosine triphosphate. These are going to be formed by our 2 molecules of succinyl-CoA. Those are those 2 ATPs coming out of our circle each time we looked at it. 6 molecules of NADH are going to be formed in the oxidation of our 2 molecules each. Remember, again, we're going to be doing the citric acid cycle twice because the glycolysis produced 2 things to go into citric acid cycle, right? So we need to do that twice, each time. Okay, and so those are the 2 molecules each of the isocitrate, alpha-ketoglutarate, and malate. Each of those produces 1. We do that twice, and so we get 3 times 2, which is 6 here. And those are NADHs. They don't actually get used up until the oxidative phosphorylation, though keep in mind that these are not actually spatially nor temporarily separated per se, though the ATP synthase is spatially separated from this. The 2 molecules of FADH2 are going to be formed in your oxidation of your 2 molecules of succinate, and again, those actually produce ATP in the oxidative phosphorylation. So again, oxidative phosphorylation, we're inside our mitochondria again, and this is where we're going to use up all that NADH. The molecules of NADH that are formed in glycolysis, each of those are going to only yield 1.5 molecules of ATP. This is assuming that they're transported via the glycerol 3-phosphate shuttle. Remember that one does that through NADH, and so it only does the NADH numbers, okay? Okay, that's the easy way to kind of remember that. The 2 molecules of NADH, these are formed in the oxidative decarboxylation of pyruvate. These each are going to yield 2.5 molecules of ATP. And so 2 times 2.5 is 5. 2 times 1.5 is 3 here. Our 2 molecules of FADH, that was in the citric acid cycle that produced that. Again, it's 1.5 molecules per each of those. And so 2 times 1.5 is 3 again. And then we have 6 molecules of NADH in the citric acid cycle. These ones will yield their full 2.5 molecules of ATP because they're formed in the mitochondria. They don't have to do the shuttle. And this is going to give you 15 ATPs because, again, 6 times 2.5 is 15. Okay? Okay. And then that will give you a net yield per glucose of 30. Now, notice these are actual publication numbers from 1991. That current 30 molecules of ATP per glucose actually will supersede an earlier value you may have heard before, which is the 36 molecules of ATP, which was the stoichiometrics of the proton pump and ATP synthase and metabolite transport. Again, these are all estimates, though, depending on your organism, depending on slight differences of pathways, depending on slight differences in efficiency. You could actually get different numbers, right? Say you had a 10% difference in performance, what would that give you? You could get like a 33, couldn't you? 20% difference in performance throughout the entire pathway, that would give you a 36, wouldn't it? Yeah. And so you can have fairly small differences, but when you're talking about large numbers like that, that would actually give you a pretty decent boost, right? So your ATP, the needs of the cell, determine, again, your rate. Oops, backwards. The electrons do not flow through the electron transport chain unless your ADP is available to be converted into ATP. The regulation of the rate of oxidative phosphorylation by ADP levels is actually called respiratory, another name for it being acceptor control. At low ADP levels, the NADH and FADH2 are not going to be consumed by the electron transport chain. The citric acid cycle is going to slow because there's less NAD plus and FAD to feed that cycle. And so you'll slow down until you need it. Okay? And so this right here shows you that ATP synthase. Again, it's going to depend upon the rest of your ATP synthesome, as well as your ETC and CAC. And so right here we again have the matrix, we have the acetyl-CoA feeding in the citric acid cycle, that NAD plus and FAD are going to regulate that cycle. That transfers electrons onto FADH, NAD plus and FAD to give you FADH2 and NADH. That feeds the proton transportation. That proton transportation is actually what ends up taking the oxygen and going to H2O in this process. That proton gradient is what drives these synthases and the ATP synthesome. Keep in mind that these are all equilibriums pressing on each other, right? So what happens if you have too much proton motive force? What's going to happen to the equilibriums that feed that? They're going to slow down, right? Because you're going to have the equilibrium start pushing backwards. Or another way of putting it is you're building up a mountain, right? If the mountain gets too high, what's going to happen when you try to climb it? Yeah, you're not going to be able to do it, right? Yeah. So the ATP synthase, this can be regulated. The inhibitory factor 1, IF1, this is an evolutionarily conserved mitochondrial protein that's going to specifically inhibit the potential hydrolytic activity of ATP synthase. This will dimerize when the matrix pH decreases. The dimers bind tightly to the ATP synthase dimers. This will prevent the beta subunits from changing conformation and thus lock, kind of like brakes, right? It's going to lock your axle, okay? This is going to be essential in cases where tissue may be O2 deprived, like a stroke or heart attack. The electron transport chain cannot generate the proton motive force. The overexpression of IF1 in some cancers is actually going to facilitate the induction of aerobic glycolysis. Non-shivering thermogenesis, this is the ability to generate heat without shivering by uncoupling oxidative phosphorylation from ATP synthesis. So this is going to occur in mitochondria-rich brown adipose tissue in animals. This is activated in response to a drop in the core body temperature. So the uncoupling protein 1, UCP1, this can also be called thermogenin, which I think is actually much better name. An intermitochondrial membrane protein that transports protons from the intermembrane space to the matrix with the assistance of fatty acids. So this right here is going to be UCP1, uncoupling 1. And so the energy of the proton gradient, which would normally be captured as ATP, is going to be released as just heat. Heat being normally referred to as waste energy. Okay? By the way, does anybody know how efficient a heater is? Electric heater? It's 100% efficient or 100% inefficient. Yeah. There is actually no difference between high or low quality heaters. Okay. So the fatty acid activated UCP1 channel again. Here we have the UCP1. It's going to release the H pluses without using them to drive the normal ATP synthesis. This would then just generate waste heat energy. Okay. Anybody mixed a stronger acid with water to make a weaker acid? No? Come on, you guys have done it before. I've made you do it before. Okay. So it heats up, right? When you go from a higher concentration to a lower concentration. It's the same thing here. You're just taking an acid and putting it in more dilute solution. And it's sharing heat. That's it. And we're, avoiding this part right here. Not doing this. So UCP2, this is the uncoupling protein that's found in a wide variety of tissues. UCP3 is the uncoupling protein found in skeletal muscles and brown fat. UCP2 and UCP3 share about 50% or greater identity with UCP1. This may play a role in energy homeostasis, may be important in regulating body weight as well. Obesity actually leads to a decrease in BAT. This right here is the brown adipose tissue, the BAT, by the way. This is pre, this is post here. And this is exposure to cold. And what you can see is the brown adipose tissue, what is it doing? It's heating up, isn't it? And so that's helping us generate more heat. Specifically, it's happening in our sort of core and leading into our brains, isn't it? So the ancestors of pigs have actually lost the gene for UCP1 about 20 million years ago when they inhabited tropical and subtropical environments. Piglets tend to generate heat by huddling with their litter mates in an insulated nest and shivering. This will lead, by the way, what time is it? Okay, I'm gonna have to use the restroom real quick, and then